Hello dear student, in this video I will talk about the process of DNA replication and transcription process. Let's start by the DNA replication. As you know that replication is the process of copying DNA molecule and making two copies of that, which it happens during the process of cell cycle in S stage, which it means synthesis stage. So during S stage the DNA it will be replicated. Okay, so for the replication we need some proteins and enzymes that they are carrying out the process of DNA replication. But before talking about the process of replication, how it happens, what are the enzymes, we have to know the structure of DNA. We know that DNA is made up of two strands, okay, and the two strands are winding around each other, or it looks like a spiral shape, and these two strands we called it leading strand and lagging strand. One of the strands, okay, let's just say this is leading strand and the other one is lagging strand. Okay, these two strands are complementary to each other and they are also opposite to each other. And we know that DNA made up of nucleotides. Okay, so these strands are made up of billions of nucleotides. Those nucleotides are adenine, thymine, guanine, and cytosine, adenine, let's say thymine. Okay, so on the next strand there will be the complementary of it. Okay, and we know that as we studied in section 8.2, adenine is always pairing with thymine, and thymine pairs with adenine, guanine pairs with cytosine. Okay, and cytosine here pairs with guanine, adenine with thymine, and this adenine with thymine, and thymine with adenine. So we have a large number of those nucleotides, really billions of those nucleotides that they are arranged on these two strands. And there's another point that we have to know about the structure of DNA. The two nucleotides on two strands, okay, on this strand and on this strand, they are joining with each other by hydrogen bond. Okay, so between adenine and thymine we have two hydrogen bond. Between cytosine and guanine we have three hydrogen bond. Between cytosine and guanine again we have three hydrogen bond. Between adenine and thymine we have two hydrogen bond. Thymine, adenine we have two. Here we have two. This is a rule that we studied in section 8.2. And what we have to know that always adenine pairs with thymine and cytosine pairs with guanine. These nucleotides are pairing with each other or joining with each other on two different strands, while the nucleotides of the same strand they are joining with each other, okay, as I am drawing in yellow color, as they are joining on the same strand is a bond that we call it covalent bond. Okay, we know that what's covalent bond. So the covalent bond, it will join the nucleotides that they are located on the same strand or single strand. Here also on the other strand the nucleotides are joined with each other, okay, by covalent bond. So this is covalent bond. And we say that one of the strands is leading strand while the other one is lagging strand. So this is the structure of DNA. Okay, we say that the process of replication is carried out by proteins. Okay, so protein they have role in the process of DNA replication. DNA cannot replicate itself. It's just it's just the place for storing the information. In order to be replicated, we need proteins, and those proteins mostly they are enzymes. Okay, as we know that all enzymes are proteins, while all proteins are not enzymes. So now let us talk about the enzymes or the protein that they have role in the process of DNA replication. Generally, we have 4 proteins. Okay, the first one is DNA helicase. Okay, helicase. As we know that this suffix ase, it means this is enzyme. The second one that we have is a protein, which it is SSB, which it means single stranded single strand binding protein. Okay, the third protein that we have, we call it DNA polymerase, which it is very important enzyme for the process of DNA replication. DNA polymerase. So this is again is an enzyme, as there is ASE, this suffix. The fourth enzyme that has a role in the process of replication, we called it DNA ligase. Okay, so we have those 4 enzymes that they are very important for the process of replication. Each one has its own function. Let us say DNA helicase, guys, what you have to know, DNA helicase, it unwinds the DNA, or we can say it unzips the DNA molecule. It means they will separate the 2 strands from each other. They should be separated. Okay, so when they are separated, as we say that they are very strongly surround around each other, these 2 strands. They are in a spiral shape. So when they are unwinded or unzipped, okay, they should stay like that in order the process of replication till the process of replication completed. So who prevent the rebinding or rejoining of these strands from each other? It should be prevented, okay, from rebinding. We have this protein, single strand binding protein, which it prevents what? Rebinding, okay, or rewinding, we can say rebinding. Okay, it prevents the 2 strands to bind with each other or to join or to wind. Here I am sorry guys, I forgot to write n unwinds. Okay, unwinds, here it's unwinds. So we have another enzyme that when the 2 strands are separated, then the DNA polymerase is an enzyme that it will come and it will add the nucleotides to both separated strands. Okay, so it will adding their function is, or its function is adding nucleotides, forming, so its function is forming new strand. Okay, this is, it adds nucleotides to both strands, especially to the leading strand, okay, and lagging strand, we can say. DNA polymerase, guys, as it adds nucleotides to the leading strand, also it adds nucleotides to the lagging strand. Okay, but in lagging strand the nucleotides will be added in the form of small fragments. Okay, after that, those fragments will be joined with each other by the DNA ligase. Okay, so DNA ligase joins, we can say, joins the nucleotide fragments in which strand? In lagging strand. Okay, so briefly guys, we have those four enzymes. DNA helicase, it unwinds, or we can say unzips, the two strands of DNA. It means it separates the two strands. Single strand binding protein is a protein that prevents rebinding of these two separated strands from each other. DNA polymerase, its function is adding nucleotides and forming new strands in leading strand and in lagging strand. But one difference that I mentioned, in lagging strand the nucleotide will be added in the form of fragment. Okay, then those fragments in the lagging strand, they will be joined by DNA ligase. Okay, so we have those four proteins that they have a great role in the process of DNA replication. Now let me show you how those proteins functioning in this process. Okay, so guys, we have this is our DNA molecule. Okay, one of the strands, let's say it is leading strand. Okay, while the other one is lagging strand. We said that. So on leading strand, guys, for example, we have let me write in red color: adenine, thymine, guanine, and cytosine. So on the lagging strand, we will have thymine, adenine, cytosine, and guanine. So we said that one of the enzymes, it will come and it will bind to the DNA molecule. Okay, let's say this is the DNA helicase. Okay, this green color is an enzyme which we called it helicase. And we said that the function of helicase, as you see, its function is unwinding or unzipping or separating the two strands. So as a result, what will happen? The two strands will be separated, guys, as you see. Okay, so on this strand, on leading strand, guys, this is leading strand, and this is lagging strand. On leading strand, as you see, we have adenine, thymine, guanine, and we have cytosine. While on the lagging strand, as you see, what we have thymine, adenine, guanine, and cytosine. Okay, so DNA helicase separated the two strands. Then we have a protein which we called it single strand binding protein. It will come and it will bind to the two strands of the DNA, and we mentioned its function. We said that it prevents rebinding of those two strands. So these proteins here, they will attach that and they will prevent rebinding. Okay, the next step, it will be the DNA polymerase. DNA polymerase, then it will come and it will add nucleotides. Okay, so the DNA polymerase, it will start adding new nucleotides. Okay, here thymine, it will be added, here adenine, cytosine, and guanine. So a new strand is going to be formed. Okay, and this process will continue according to the length of DNA. On the next strand, again, the DNA polymerase, it will add fragments, okay? Fragments like that. For example, here it will be adenine, here it will be thymine because we have adenine, we have guanine, so it will be cytosine, and here it will be guanine. So a new strand it will be formed. And finally another enzyme it will come, which we called it DNA ligase. Okay, DNA ligase, it will join the fragments with each other. Okay, so finally, as you can see guys, what you can notice from one DNA molecule, okay, in the same sequence, adenine, thymine, guanine, cytosine, okay, two molecules formed. Okay, this is the first one and this is the second one. Okay, with the same sequence, adenine, thymine, guanine, cytosine. Here it will be again adenine, thymine, guanine, and cytosine. So guys, as we can see, we have two copies of DNA molecule. And one important point, the process of replication, we can say it is semi-conservative. Semi-conservative. Okay, what we mean by semi-conservative? In each one of the two DNA molecules, there is one old strand. Okay, this green color is our old strand, and we have a copy of that strand in the second DNA molecule. That's why we call it semi-conservative. Okay, there's another point I want to mention. You know that the DNA is very long, guys. It's very long. It's made up of billions of nucleotides. So the process of replication, in order to be fast and accurate, okay, in order to replicate the whole DNA molecule, that long DNA molecule, in order to be fast, we have many origins of replication, okay, in the DNA. Origin of replication, or we call it ORI, okay, ORI. We have many of those origin of replications in the DNA. What's ORI? So guys, is the starting point, we can say, of replication process. Okay, but we have many of those ORIs in the DNA. So the process of replication, it will start from the many points. Okay, that's why it will be very fast. The process will be very fast and it will be very accurate. Okay, that's about the process of replication. In the next slide, we will talk about the process of transcription. What's the transcription process? So guys, we have the process of transcription. The process of transcription, it means taking the information from the DNA, or we can say converting, okay, converting the information that is stored in the DNA, converting the informations, okay, converting the information from DNA molecule into RNA molecule. As we know that RNA is a ribonucleic acid that differs from the DNA in 3 points. We mentioned that, which they are, in which we have ribose sugar, okay, and it's single-stranded, the RNA molecule, and we have uracil instead of thiamine, okay. But the most important point, we have to know that the RNA molecule is single-stranded molecule, okay, while the DNA, it is double-stranded molecule. So you know that, guys, DNA is like a cookbook. Before we talked about the DNA, it contains all of the informations of our body. So all of the cells in our body, they contain the same DNA, okay, but according to the need of that cell, according to the structure and function of that cell, a group of gene, it will be transcribed. As an example, let's just take our pancreas, okay. Let's say this is our pancreas, okay. So guys, the pancreas is responsible for releasing a hormone which we called it insulin hormone, okay. We have this hormone which it has a great role in regulating the blood sugar, okay. So guys, as I told you, all of the cells or all of the organs of our body, they contain the same DNA, but they will transcript or they will convert the informations from the gene that they need. For example, our pancreas, as it needs insulin to produce insulin, the gene of insulin, it will be transcribed, while this gene will not be transcribed in our like skin cell, let's say, okay, because our skin cells do not need insulin hormone. Or let's say if we take a cell from our mouth, okay, let's say this is a cell, okay, that it contains a nucleus. Inside the nucleus there is DNA. Which gene it will be transcribed, guys? For example, we need amylase, okay. This is an enzyme that's produced in our mouth, but amylase, so the gene of the amylase, it will be transcribed, but it will not be transcribed, for example, in our lung cells, okay, because we don't need it in that place, okay. We have that gene in our lung cells, but it will not be transcribed. So I hope it's clear what we mean by the transcription process, okay. And now let's us see how the process of transcription, it will happen, or how it will process it. We have an enzyme that we called it RNA polymerase, okay. RNA polymerase is one of the most important enzymes for carrying the process of replication, that has a great role in the process of, sorry, not replication, guys, transcription, I mean. So now let us see how the process it will happen. So we said that we have the DNA molecule, okay. This is our DNA, which is very long, okay. In a part that we need that gene to be transcribed, that place it will be unwinded, okay. As you see, this place, guys, as I did by myself, it is unwinded or unzipped, okay. For example, it contains a gene which it is insulin gene, okay, because our body needs that gene, it needs that hormone, okay, for regulating blood sugar, okay. So in order to be transcribed, at first we have a group of protein that they will come and they will bind to this place which it contained that gene. We called it transcription complex, and it includes RNA polymerase enzyme. So this is a protein or an enzyme that it will bind to that place of that gene, Okay, so this is, we called it transcription complex, which it includes, guys, this transcription complex is a complex of many proteins, and the main protein that it includes is RNA polymerase. Okay, so this transcription complex, it will invent the DNA molecule. Okay, so if, let's say, if we have, again, we will have adenine, thymine, guanine, and cytosine, and also cytosine, guanine, adenine, and thymine. Okay, here, so we have what on the strand? We have thymine, adenine, cytosine, guanine, cytosine, thymine, and adenine. Generally, the DNA strand is made up of those nucleotides. The RNA polymerase, when it wants to transcribe that gene, it will use, this is very important, guys, pay attention, it will use one of the strands. Okay, it will use only one of the strands, and it will start by reading that strand, the sequence of the nucleotide, and it will form the complementary of it. And as I told you, in RNA we don't have thymine, instead we have uracil. So guys, adenine, it will pair with uracil here. Okay, U. Thymine, it will pair with adenine. Guanine, it will pair with cytosine. Cytosine, it will pair with guanine. Here also we have guanine, cytosine, adenine, it will pair with, we don't have thymine, we will have uracil, and thymine, it will pair with adenine. So as a result, a single strand of RNA, it will be formed. Okay, when the gene is completely transcribed, then this RNA molecule, okay, this is RNA molecule, it will be attached or detached from the DNA molecule, and it will come to the outside of the place. Okay, so that RNA, it will be separated from that DNA. Okay, so we will have uracil, adenine, cytosine, guanine, cytosine, uracil, adenine. So this is our RNA. Okay, that's formed, and it is separated, it is inside the nucleus. So as a result, when this RNA, okay, is removed from this place, let me clean it, guys. As you see, it's detached from the DNA molecule. Okay, it comes to the outside. That's very important for us, that we have to know this RNA. Okay, then again, guys, the unzipped area here. Okay, so after that, just I want to tell you, this transcription complex, it will be broken down. Okay, it will be removed from that place. It will be, this complex, it will return to its main component. So many small components will be formed of that transcription complex. Okay, that they made up of RNA polymerase and many enzymes. Okay, they will float inside the nucleus, and as a result, guys, what will happen? Let me draw it here. The RNA, again, the DNA, sorry, it will be winded around each other again, and the RNA that's produced. Okay, as we say that the RNA is produced. So the RNA, guys, it contains the informations, okay, that we need. When it's formed, it will move to the outside of nucleus. So it will move into the cytoplasm. And there, inside the cytoplasm, the process of translation will start. We will talk about the process of translation. What is the translation process? Okay, so these three processes, guys, we can say replication, and transcription. This is transcription, and translation are regarded as the central dogma of molecular biology. Okay, the last important point that you have to know, replication and transcription occurs inside the nucleus. Okay, while the translation process it will happen inside the cytoplasm. This is for a eukaryotic cell, while for the prokaryotic cell, as you know, we don't have nucleus, so all the three processes will happen inside the cytoplasm. So that's about the process of replication and transcription. In the next video, we will talk about the process of translation.